Hi, everybody, and welcome to the ROSA Worldwide Webinar Series. My name is Mike Reed. I'm the Chief Scientist at ROSA, and I'm your host for today's webinar. Please check the ROSA website for upcoming webinars. We have them about once a month with modeling leaders in the field. Our speaker today is Jeff Kearns from Novartis Biomedical Research, where he co-leads the Translational Modeling and Simulation Group. He held previous positions in small pharma at Forma Therapeutics and Merrimack Pharmaceuticals. At Novartis, his portfolio spans cardiovascular, metabolic, neuroscience, and renal disease areas, and both traditional and novel modalities. In recent years, Jeff has had specific interest in RNAi and has held several modeling and company leadership roles to expand the development of the Novartis RNAi platform. Please join me in welcoming Jeff Kearns. Okay, take it away, Jeff.
All right, great. Thank you, Mike, and also Heather, for the invitation and the kind welcome. Um, I am pleased to actually share the work of both my own group and also others. Really giving a state-of-the-art lecture on where we are in the RNAi field, both at Novartis and in the rest of the pharma field. So just for my disclosures, I'm an employee and a shareholder, and my wife is at Sanofi. In terms of the agenda, so I have broken it into three parts. In the first third, I am going to give a state of the art, sort of what are the principles of the modeling of the PK and the PKPD for our RNAi drugs. What are the principles, what are the models, what are the data that we often have to use. I'm then going to try to bring up those principles in a series of the vignettes. I've got it scaled for time, so if I am running long, I might actually skip at the last one. And then I'll end with on a perspective of where I think the field's actually going next. All right, so just as a blatant plug for our group, we are the translational M&S team. We are in the biomedical research part of the company. Right now, we're at 29. I think it might be 30 as of next week, with our primary locations in Cambridge and in Basel. We are actually part of a broader group that includes non-clinical statistics, a PBPK group, and a data science group. The modeling approaches that we apply are the standard ones across the field in terms of PKPD, PBPK, PD, QSP. And we work across all of the disease areas and the modalities from the standard small molecule or antibodies up through siRNA, radioligands, multi-specific cell and gene therapies, and more. And we're still growing, so if you want to check our web page, I think we have some of the positions up there now. All right.
So,
So
Jeff, just a quick comment. If people want to enter questions, there's a Q&A button at the bottom of the screen, and we'll address those at the end of the talk. Sorry to interrupt.
Yep, will do. All right. Yeah, so the state of the art, and I have set this up as what do we need to know to be effective in the RNAi space. Um, so a very quick primer with apologies to all the biochemists and the chemists who might be in the room. This is very brief. So there are multiple approaches used across the field for actually repressing or knocking down expression. Two of the more canonical approaches are ASOs, which are single-stranded, and siRNA, which are double-stranded. You can see this is actually from a recent review article, 2023. There's a host of drugs that are either approved or in clinical development now. There's even more that are on the list here. For the topic for today, I'll be covering the siRNA space. That's a little bit of a cleaner topic. And then also apologies for bringing up another term in our field of acronyms. At Novartis, we are actually referring to these drugs as xRNA. So it's a little bit of muscle memory. So that's what I'll be referring to today. In terms of how we think of this from a modeling approach, right? So these are often a modular drug that are consisting of a ligand, a linker, and a payload, which is really informing a sum of parts approach, where it's the ligand actually driving the PK and the bio-D and the cell binding and the cell uptake, and it's the payload and the linker that are actually driving stability within the cell and the long-acting PD. Even if you are actually coming out of the ADC field, this actually looks very familiar. Just replace the ligand with an antibody. Right now, the bulk of the field, or where there's the most advanced parts of what the field, are in the liver xRNA space that all use the same GalNAc trimer as the targeting ligand. But there's also a wide array of emerging approaches that are outside of the liver that are applying other ligands such as Fabs and IgGs and fatty acids and more. All right. So in terms of the principles, we have a team of the modelers here that are working with project teams. These are the principles that they often approach and need to work with their teams on. So the first one is it's the tissue PK/PD that actually matters most. And if you're coming out of the more traditional spaces, small molecules and mAbs, this is going opposite of what you often have to think about. So, while plasma PK has its utility for looking at safety margins and tissue delivery, it's not really a good predictor of xRNA potency and the durability. On the other hand, it's the tissue PK/PD, which is the important driver here. And so that might require the measurement of total drug levels within the tissue. That might include also loaded risk. Um, Oftentimes we are lucky, and there's a secreted protein marker that actually acts as a pretty good surrogate measure. And it's really critical, especially as you move outside of the liver space where the delivery isn't as efficient, really having a good modeling understanding of tissue PK/PD. Principle two is that it's actually possible scaling from preclinical species to man, especially in the liver xRNA space. So rodent or transgenic rodent to monkey to man is actually a pretty straightforward process. You can also scale tissue PK/PD, but just like any species scaling when you're dealing with a process within the cell, you just have to think about, are you scaling by metabolic rate, or what are the nuances of that? But it is possible. Most of the time, you and your project teams are going to want to know, how does the potency, how much knockdown am I getting? How's the durability? So how long does my effect last? How does that translate from mouse to monkey to man? And you have to consider a host of effects in terms of stability of the drug within the cell over weeks to months to years, what the target levels are, and the turnover rates. And really, this is the core part of the strategy for any anticipated human dose that you would apply the model for. Principle three, and we need to remind ourselves about this often, timescales matter. The first principle is sort of hurry up and wait. So on the delivery side, so that's your, administration, often subcutaneous, sometimes IV. That's fast and it could vary. It could be very efficient, or in the case of non-liver targeting drugs, very inefficient, but it's very rapid. It could be hours to days. On the other hand, this waiting period, your effect is it's very slow to have an onset time. It could be weeks before you hit peak knockdown levels, but it's very durable and can actually persist over months to even a year or more. Um, the other thing is that timescales are driving the tissue bioavailability. And as I said, it's not the plasma PK that matters, it's the amount of drug that you can actually deliver into the tissue that drives the effect and the duration of that effect. But the delivery is actually saturable. And so you really want to avoid any systemic PK that will actually saturate any capacity of the cells to internalize drug. If you're coming out of the biologics space, this is basically a TMDD inflection point. And rather than actually trying to saturate that, you want to just hit it and not go much further. And you can achieve that by actually recognizing that the dose is a tunable property and that you can change the dosing route or the dosing formulation and reduce the Cmax and move your exposure into the terminal phase so that you don't actually saturate cell uptake. In terms of the kinds of models that we use, there really are two. So you have your KPD model, which is more of an empirical approach. This is a very straightforward model to use. If you're coming from other modalities, you probably use the same model. So this just relates administration of the drug goes into a drug compartment with a clearance rate on it, and you have an Imax term on the synthesis rate of your target. This is an empirical approach, but there are fewer parameters, and oftentimes it's limited to when you have some data. Um, there's the more mechanistic approach, and we've seen numerous examples across the field. Shown here is an adaptation of a figure from a 2021 paper in which Viva here made a PPK model that includes plasma, liver, rest of body, and the renal clearance. It is a mechanistic model, so it has receptor binding in the liver, so you're delivering cells into hepatocytes, at which point drug actually sits in a late stage endosome over the course of weeks to months or more, and has an escape rate. It loads onto the RISC complex. It binds to the target mRNA and leads to the degradation of that. It's got extensive utility, especially early in lead optimization, but then also later when you might be changing the dosing route or the dosing formulation in a way that wouldn't be compatible with your more simplistic KPD model. This is a more text-based slide about actually knowing what kind of data that we have to put into these models and what models that we might want to use. So, an early decision that you might have to make is which of the modeling types is most appropriate for the project and for the project stage. And so we're oftentimes thinking, is it more appropriate for the mechanistic PBPK-PD model? Do we have data on plasma and tissue PK-PD? Do we need loaded risk? Are there any cell-based assays that we have to bridge to? We try to use the KPD model if that's actually sufficient in a more fit for purpose manner. In terms of the in vivo studies, a lot of what our group has to do is work with preclinical teams on study designs. And because you have cross-reactivity needs for both the targeting ligand as well as your siRNA sequence, we are often thinking, is a rodent an appropriate model? A transgenic rodent that expresses human target is oftentimes used. Monkey, it might cross-react with your drug, but most often not, or might have reduced potency. So those are always things that we have to think about in our study designs. In terms of what kinds of studies that we actually run, and you'll see some examples of this later, if there are questions about potency, so how much knockdown can you achieve? What's the time to effect, which can often be weeks after administration of the drug, or what's the BioD into your site of action into off-target tissues? We will recommend a single dose study, oftentimes of a shorter duration. If there are questions about durability that really lead to how often would we have to administer this drug, there we're now talking about prolonged washout studies after either a single dose or multi-dose where you can achieve steady state, and then you look at the recovery phase. And then it's also really important to include controls if you actually have them in terms of other front-runner compounds or ones that you have any clinical data on that you could actually use as a translation path. Um, just like in the ADC world that we're experienced with in the oncology space, the bioanalysis methods are actually a real roadblock to how our modelers have to interpret the data and what studies that we can recommend. You just need to get familiar with what are the standard data types. Is it an MSD assay or stem loop RT-PCR or LCMS MS if you're looking at metabolites, many of which are actually still active. So there's that's a whole separate talk, more of a DMPK talk about what are the bioanalysis methods that you would actually have to look at. Um, this is a more recent one, is to sort of know the toolkit and know the space. So there is emerging guidance. So this here is a draft guidance from the FDA from the end of last year. It's a recommended read. I've got the screen grab at the top right. One of the more interesting parts of this guidance from a modeling perspective is this recognition that if we are claiming that PK and PD are uncoupled, so you have rapid plasma PK, but a prolonged PD effect later, that if that's what's happening for efficacy, that could also be occurring for your chronic toxicity. And so incorporation of PD dynamics within the design of your non-clinical safety studies is actually an important criteria called out in this guidance. So it isn't just trying to run a non-clinical safety study to actually determine what's your MTD, what's your no AEL level, what's the Cmax and the plasma exposure at those doses. Because your plasma PK and your effect are uncoupled, that might not always be the best approach. It's a great opportunity for your modelers working with the non-clinical safety and the in vivo pharmacology teams on what are the appropriate study designs. The last principle, and then I'll move on to the vignettes. The more recent addition, as we've seen an explosion across the pharma field in the academic spaces, that there's a lot of other opportunities for targeting ligands. And from a modeling perspective, if we take a sum of parts approach, just because the siRNA payload is new doesn't mean everything is new. I think the clearest example of this is in the so-called AOC space. So these antibody oligonucleotide conjugates, they're essentially they're an ADC with a non-traditional payload. And what that means is all of our modeling methods that we have learned primarily in the oncology space for ADCs, most of those apply. So all the plasma PK, the BioD, the receptor binding and internalization, linker stability, all of those, all of those things that we've actually had to learn how to model in the course of the last 25 years apply here. It's just the PD component after the cell is actually delivered. That is the new part of this model. And we're seeing that in other spaces also. We have examples of a fatty acid siRNAs, kind of older school, but we have our nanoparticles are still being used. There are multiple approaches for the tissue targeting, and we've got models for most of those. And so to sum it all up, I just grabbed a pretty common image that we oftentimes see when you have a lot of complex data. It often feels as a modeler on these project teams that we're actually flying the airplane as we were building it, and we're adding on parts about plasma PK and dosing routes and tissue BioD and long-term PD effects. And that really it's the modeling acts as the integration hub for all these activities and really helps us inform teams. Okay, so with that, I'm going to jump into some vignettes with apologies that most of these have actually had to be blinded, but what the targets are is less important than what we are actually learning from the vignettes. So starting from very early stages, so you're a modeler on a team that might be at the project start stage or has just come into the lead optimization stage. What can you help your teams with? So a scenario here. So you are called in to help a team who is developing a next generation drug with the aim for longer durability. So maybe the drug in the clinic right now is administered once every 3 months, and they want to administer the new one every 6 months, or it's 6 months and they want to get to a year. Those are common. Those are common targets, at least in the liver xRNA space. So in these scenario here, you have clinical data on a front runner that is administered on a more frequent basis, and you want to know what improvements would you have to make to that drug to enable the new TPP. And so what this might look like is a team comes to you and says, hey, our front runner drug is Q6 month. What do we need in the next gen drug to have it be once each year? And so this is really the power in early projects start times where you don't have any data on your compound yet. It might be an idea at the marker board. You can use toy model simulations. So here our standard workflow is we fit the KPD model to the clinical PD data on the front runner. So that's a pretty simplistic model with just an IMAX term, and in IC50. We'll then run simulations of that model looking at potency and the durability. We'll find the conditions that actually meet what the TPP is, and then we'll work with our chemistry teams. Because it might be that you've actually saturated on the potency, and any gain that you would actually expect to have would actually have to come from the durability or vice versa. And so this is actually from one of our projects. I just need to blind let the target where we fit the clinical model. So on the y-axis is your target knockdown. On the x-axis is how many days. I really want you to appreciate that that actually is days and that's not hours. That is the power of these siRNA drugs, where they're incredibly durable once they are delivered into cells. They sit in a late stage endosome for months or more, and you have a a PK effect, which is gone within 24 hours, and a PD effect that can last months to years. So in the case here, this red line is our clinical front runner, which is actually dosed once every 3 months, or in the case here, once every 6 months, sorry. Um, and the team wants to know what would you have to improve. And here we could run the model showing that if you improve the potency 4 times, you'll improve the potency, the level of knockdown. You'll extend the durability a little bit, but not enough as to what the TPP requires. You could then improve these. You could improve the stability, which could be adding chemical modifications onto the siRNA payload that actually protect it when it is within the cell. And there, you get a little bit of both. You get a little bit more durability, but not enough. And it's really that for this particular target, you have to improve both the potency and the stability for it to actually achieve once every year dosing. Another scenario common in the lead optimization phase is your team is actually generating their candidates. They're taking their hits and they're putting them into in vivo studies. They're freaking out a little because they're either running hard to run transgenic rodent models or very long cyno studies, and they're measuring PD knockdown potentially in tissue with repeated biopsies. These are complex studies. They're generating a lot of data, and they want to know, are we there yet? So have we hit the durability in a transgenic rodent study that would be translatable to the dosing interval that they might need to hit and the potency that they have to hit in a clinical study. And so how we approach this is essentially the question is, what does my in vivo data have to look like? So like, what is the success? What does the goalpost look like? And so for here, what we have found through repetition across the whole portfolio again and again is that reverse translation of an idealized compound is a very useful strategy. And so you basically start with a model simulation of what you would have to see in the clinic. So you actually create a a human PKPD model of an idealized compound that actually hits the potency and the durability that you need. And because of the fact that we can both forward and reverse translate with both the KPD models, if we have some front runner data, or the mechanistic PBPK-PD model, if we don't, because we can both forward and reverse translate, we can take that idealized human PD profile, reverse translate it back to monkey or to transgenic rodent, and use that as a goalpost. So in the case here, we had a project team that was early in the lead optimization stage, so they just had their first few hits. This was long before they found their candidate. And you have a case where this black line is the reverse translation of the idealized human drug that would match the TPP. We reverse translated here into monkey, and the red data in the model fit of it are from one of the early hits. And we could actually meet with the project team and say, you've probably achieved enough potency, so your knockdown is within range. It's really now you have to focus on the siRNA stability, and so that informed the selection funnel and their remaining hit to lead strategy, where they then actually focused more on durability. But actually having a plot where you can say this is what you have to see for this to be a good drug really helps here, and it is model informed. So then as you move forward, so my last two sets of the vignettes are more preclinical to first in man, and then a little bit past that. Really, once you have that lead, you then actually have to sh shift the thinking more into the dose and the dosing route. So you'll work with your manufacturing group on how much drug supply. You'll work with your formulations team in terms of, is this an IV or a sub Q or a slow release sub Q or a localized delivery, and you need models of that. And so you have a scenario here. So I am setting this up as, you're a modeler coming from the small molecule or the antibody spaces. And we often think of this kind of question as like, well, I'll use an IV dose because it's fast, has 100% bioavailability, but recognize that it's less convenient for the patient and might not meet the TPP. Or you might say, well, a subcutaneous dose is more convenient, but probably is going to need a higher dose because we'll have slower absorption and especially lower bioavailability. So we have to dose higher. So these are the preconceived assumptions that we oftentimes have coming from these more traditional modalities. The question is it also true for liver xRNA? And the way I ask the question, you should assume probably not. And so it then comes down to, you know, we have a toolkit. So we have our PKPD model. Um, what can we learn from that model in terms of where does the drug go and how is that affected by the dose and the dosing route? And I want to call your attention to, on the right-hand side, we basically have two parallel clearance routes in this kind of model. The first is renal filtration. And so it's really driven by the renal clearance and the GFR. And then the other is just the uptake in the liver. And so the on-target receptor binding to the hepatocytes, the ASGPR on the cell surface of the hepatocytes, like, actually is a clearance route. It's an intended clearance route, but it removes the drug from the circulation. So it's really these two steps are in competition, and the role of the modeler is to figure out, for that target, for that TPP, what's the optimal dose and the dosing route that actually balances this trade-off of clearance through the kidneys and the saturation or the engagement of ASGPR on the hepatocytes. And so what this ends up looking like, I'll just show a case study that you can repeat in your own hands. This is using published data. As a class effect, we have seen the same behavior across our own internal compounds. We've seen it in the literature as well. So this is a very relevant class effect. So here I'm going to use um, phase one view Tisserand data. So this is a GalNAc siRNA, against for the treatment of transthyretin mediated amyloidosis, TTR. This was a single dose phase one SAD, from 5 to 300 milligram, where they measured plasma PK, urine PK. So that's our renal excretion, and protein PD, which is a surrogate marker for our liver uptake. And the data was actually published in this paper here, cited back in 2021. And so this is what the data look like, and we fit the model against it for plasma PK, and then these two parallel clearance routes for both urine PK and the target knockdown. Um, Oop, I went the wrong way. All right, so now that we have this model, we can then use it. And so we can compare an IV versus a subcutaneous dose. And what we can see at the very low dose, this is well below what you actually need for efficacy. You can see the y-axis here is target knockdown, so you actually want to go lower. And that In the case here, the 5 mg dose, you have a superior knockdown profile for your IV dose versus the subcutaneous. When you go a little bit higher to a 14 mg dose, which is also well below the efficacious dose, now you've got an equivalent knockdown curve. Both of them have a time to effect of about 9 to 10 weeks. And then once you begin to get up towards the efficacious dose range, now it's actually flipped the other way, where you have higher potency and slightly enhanced durability with a subcutaneous dose. Um, and so we have this mechanistic model, so we can ask why. So we can reduce this down to first principles. And the first thing that we actually looked at is, are we saturating the liver uptake? And the answer is yes. So on the left-hand side here, this is just a plot of free receptor. And so in the absence of drug, it's at 100. And you apply an IV dose, and within minutes or tens of minutes, you've completely saturated his GPR for a period of about 3 to 6 hours, in which you've now got wasted drug. On the other hand, with a subcutaneous dose with a time delay due to the absorption effects, you are actually hitting about half of the ASGPR and not actually saturating it. And what that means is, when you look at renal excretion, so if you modify the model to add a tracker species on the renal excretion, what you can see is that you are actually losing approximately 80% of your IV administered dose to the renal clearance. Whereas with the subcutaneous dose, you are losing about 20 to 25%. So your subcutaneous dose is more efficient. And what this means is, if you then look on the right-hand side, this is liver uptake, and you can actually see that you have higher drug levels with a subcutaneous dose. And this is actually supported by the data. I just grabbed some random papers looking at other siRNA drugs for the liver, where you see superiority of an IV dose at lower doses. It flips the other way at the higher doses, where you might have higher plasma PK with your IV dose, but lower drug levels in the liver. We see this for yet another compound here, where your IV dose here in a monkey study is actually inferior to subcutaneous dosing at much lower doses. So the key understanding here is just to really have a good understanding of where your drug is going and the BioD and the competition of delivery and clearance. I'm going to quickly hop through this one and just hop onto the last vignette here, which is more clinically focused. So this is inferring target biology, bridging between phase 1 and phase 2 patient populations, with really the question of, if we see this in phase 1 healthy volunteers, what might it mean when we go into at-risk patients in phase 2? So the case study here, for which there are multiple drugs in clinical development now, is lipoprotein A. It's a cholesterol carrying particle where higher levels are associated with ASCVD risk. There are multiple ASO and siRNA drugs going after ApoA, which is a component of that particle. And so again, the scenario is, how do you bridge between phase 1 healthy volunteers who have lower levels of LPA and phase 2 at-risk patients who have higher levels? A great case study of this. I would recommend reading both of these papers. You can digitize the data and take a look at it. So this is for lepidisiran. So these authors, showed the phase 1 and the phase 2 trial results. Phase 1 healthy volunteers SAD, phase 2 repeat dosing in an at-risk population. And what they basically saw was, in phase one, where you had healthy volunteers, you had higher durability because they had lower levels. In phase two, a higher dose intensity, either a higher dose or repeat dosing, was actually required in these at-risk patients. And we wanted to see if we could explain that from first principles, just actually looking at the changes in the LPA levels. And so short answer is yes. So we basically fit the model to the phase one data, where they had lower LPA levels. And then we did a dose titration or sorry, and we did an up dilution of the synthesis rate, or up titration, sorry, of the synthesis rate, moving from what you would see in healthy volunteers to what you see in the phase two. And what we were able to actually show, is that we could do a pretty good job predicting the phase two response in at-risk patients using just the phase one data in the healthy volunteers. Um, so I'm going to end in just these last minutes with what's next and where is the field going. Sort of a brief perspective. I think the first thing that should be clear for anyone who's in the siRNA field now is that there's a rapid race on to unlock the next tissue. I think as a modeler, we're hugely advantaged in the liver xRNA space because we're all using the same ligand. We know the PK, we know the PBPK. As we expand past the liver into other tissues where we're changing the ligand, we're changing the targeting strategy, there's a real need for us to improve the modeling and to bridge to current understanding and historical understanding with those approaches. Really, the name of the game is coming up with good analysis methods and study designs and dosing strategies that will maximize tissue bioavailability. There's a real need for model-informed approaches both to the dosing route and the formulations. What I didn't have time to show is that middle case study where you could actually compare an IV, a subcutaneous, and a slow release subcutaneous, and showing what the effects were there. Um, More of these targets outside of the liver are exploring sub-maximal inhibition. So you're not just hitting everything with a sledgehammer and trying to knock down as much as you can for as long as you can. You might be hitting 30% knockdown or 50% knockdown, and now you need modeling approaches to look at reducing the peak to trough effects of a PD knockdown around a midpoint and not just trying to drive it into the ground. Um, more future looking, all these chemical modifications that are being made to these siRNA drugs are really impactful on both the potency and the durability. And this is a hugely combinatorial complex space where we simply don't have the data yet to inform a machine learning approach. But right now, that actually forces us to be empirical with what is the effect of this new chemical modification or this precise profile of different chemical modifications. So there's more work needed as the field grows. And then also as you saw in that FDA guidance, that we need to incorporate our PBPK/PD or our KPD modeling into informing study designs, that are shorter in length in the context of long-acting compounds. Um, and just to end with that, right? So just this idea of moving past the liver on the left-hand side is just some press releases on one of our approved drugs in the liver xRNA space. So this is our Leqvio drug, or in glycerin. There's a whole field following behind us. Like liver delivery from a modeling standpoint and from a development standpoint is pretty well understood. On the right-hand side, this is an adaptation of a news piece out of the biospace, looking at the sort of variety of different targeting strategies and approaches that are actually targeting outside of the liver. So whether it be um, in brain for tau, in the lung for asthma, skeletal muscle for DMD, DM1, and others. There's a lot of work that is being done with new targeting approaches. And so this requires some more expanded modeling. Um, and I'll skip through that. And I just want to end right on time with the acknowledgments for these are the six folks on my team who have been doing really outstanding work. From ideas on a marker board up through life cycle management on our approved drugs, really applying novel approaches for modeling across all of those project stages. Of course, other associates within our broader department and the project teams and these stakeholders. And so with that, I'll end right on time and happy to answer questions, which I can't see the chat while I'm in slide view, so I'll need to rely upon Mike.
Thanks, Jeff. Great talk. Really appreciate it. I have a few questions here that we'll get through as many as we can here before the
Certainly.
top of the hour. First question is, how do you translate the PK of siRNA, I think from preclinical to human predictions? Is it allometric scaling, or can you speak
Yeah,
a little
so
bit about that?
so I think it actually comes down to a more straightforward question is it's not about the siRNA. It's about so most of the drugs are linked to something else. So for the liver siRNA drugs, those are predominantly all linked to a GalNAc trimer that engages the ASGPR receptor on the hepatocytes. Other approaches are using antibodies and Fabs. So the PK scaling is traditional approaches for scaling PK for that particular targeting ligand.
Gotcha. Yep, makes sense.
So it's oftentimes allometric scaling with a body weight and an exponent. Um,
So when you have, or when other groups have done actual human measurements, does that seem to bear out in terms of uh, when an siRNA is attached to it, it behaves similarly to what has been reported previously? Do you ever
that
get any
has been,
surprises?
yeah. So our track record so far, and this includes both our robust internal portfolio, as well as, we scour the literature and we fit models to what's out there. Our experience has been that it follows the targeting ligand, which is very similar to, I spent years in the oncology space working on ADCs.
Mhm.
Often, unless you had some crazy payload, it often follows the targeting ligand.
Great. How do you model the transport of the drug from plasma to the tissue?
So we have two approaches, right? So in the simplest approach, the answer is we don't model it. So in the KPD model, we have a drug compartment and an indirect response model with an Emacs model. And there we are completely uncoupling PK and BioD in cell delivery, and we're just modeling it as an Emacs model. In our more formal PBPK-PD, a in our more formal PBPK-PD approach, we are modeling the BioD of the targeting ligand, then the receptor binding, the internalization, and all of that's finished within 24 hours. And then the rest of the model for the next months to years is all the PD effects within the cell.
Mhm. Great. What are the analytical methods used to measure the risk and the xRNA? Are there methods
Yeah,
more
so
suitable for reliable modeling?
I'll jump back. Um, Jump really far back. Where were we? Okay. So the bioanalytical strategies are still emerging. I think most often for plasma peak for the plasma PK, we have a hybridization MSD or a stem loop PCR, which could also be used for the tissue measurements. All of these are pretty standard. You include controls and a standard. So at least for modeling purposes, these are pretty fine to use. I think where we're going is also recognizing that there are active metabolites. So these are siRNA drugs that are often a 21 to 23 mer, and you have active metabolites, so your N-1 or your N-2 metabolites that are truncating bases. And those are often still completely active, similar potency, if not like the same. And for there, we're now needing to expand the toolkit for modeling purposes of really looking at total active payload, which would include the parent siRNA as well as the N-1 and the N-2, for which you need a mass spectrometry assay or a well-calibrated stem loop RT-PCR. Sorry, that was a long answer, but it's a little bit of a passion area. Mike, I
Thank
think you're
you.
on mute.
Yeah,
Yeah.
yeah. Could you speak a little bit more about the distribution studies and translatability across species? And really, the goal of this question is getting at, can we eventually replace in vivo studies with, in vitro and modeling to get to some of those translations?
Yeah. So um, I'll address that as sort of a sum of parts, right? And I'll start with the PK and then I'll get into the PD. So for the PK, what we oftentimes hear, and again, I'll just go to the liver space because that's where the bulk of the field is now. Um, um, they're all using the same ligand, right? So they're all using the same GalNAc trimer. And the BioD and the PK by are predominantly driven by GalNAc. And so one of the common questions that we hear is like, well, do we need to measure PK for the new drug, or could we assume it from all that we've measured on past drugs that use the same ligand?
Hmm.
Can we avoid running an in vivo PK study? Um, the answer is maybe, right? So you certainly need it for safety. You want to include it in your preclinical package, so you can't completely avoid it. But what we have been seeing is that the PK for all of our liver xRNA drugs are all within the same variability that you would expect seeing other liver xRNA drugs. So you might be able to in the future avoid running PK studies. What the PK also tells you is that if it's not drastically different from other liver xRNA drugs, that probably your BioD and your liver uptake is also probably pretty similar, if not the same. At least within your precision of your measurements. So there's a lot that we can infer about the next drug if all we know is the dose.
Hmm.
Um, it's nice to have like the studies in terms of knockdown, like what happens within like the cell, right? So these are your PK is done within 24 hours. Your PD is months to year. Um, we have in vitro or cell-based assays looking at the cell stability of the siRNA once it's actually within the cell, the escape rate out of the endosome, the risk loading and the target knockdown. We're having trouble having a reliable way of bridging straight from biochemical or in vitro or cell-based assays into in vivo. It's okay for rank ordering things in the lead optimization phase, but it's not always directly translatable. And so you wouldn't be able to avoid the in vivo study there.
Okay, thank you. Could you speak to the potential for modulation of the delivery receptor dynamics? So, for example, turnover rate that might occur based on treatment or could be different between healthy volunteers and disease population. Does that ever play a role in the modeling of these
So
processes?
that's a great question. I think the first part of that question is saying, is delivery a time varying effect? And um, Right now we haven't seen that. At least with the targets that we are looking at, right? So some of this might have to do with the fact that you are administering drug once every 3 months or 6 months or once a year or whatever your longer dosing interval is, and whatever transient effects that you had on receptor levels are mitigated or lost before you come in with the next dose. So you might have pushed the system into a refractory period where you have lower receptor levels just after dosing, but by the time you come back 6 months later, that's not present anymore. So where it might come into play is fixed dose combinations where you have more than one product, or if you're administering more than once within a short period of time, there we would skip over the KPD model, go right into the minimal PBPK-PD model where we have the receptor trafficking there.
Great, thank you. Uh, I think we probably have time for about three more questions. First one
Okay.
is about preclinical species that is most predictive of human, uh, PK and PD. Have you found that NHPs are more directly translatable to the human outcome than other preclinical models?
Um, I would like that to be the case. I think we would all like that to be the case. Um, I think that might I think that might end up being true, at least in the liver xRNA space, if you have a transgenic rodent that expresses human target. Because again, this goes back to complementarity and cross-reactivity for both your targeting ligand and for your siRNA. And just even having one or two or three mismatches between your drug product and in the mRNA in a monkey might be enough to actually throw the potency off.
Mm,
Uh, and so having a transgenic rodent where you have human target, where you have the same potency helps. I think this is a more retrospective question where I think we need enough examples within literature to like where you have transgenic rodent, you have monkey, and you have phase 1 clinical data, and you could do a retrospective analysis and say, did I need multi-species scaling, or could I have accomplished this just from the transgenic rodent? I can't speak to our internal projects, but that's certainly on our radar, and we're doing that.
okay, thank you. What about siRNA uh, therapeutics that are intended for the CNS?
Yeah,
How,
so
how are they typically delivered? This question.
yeah, I mean, I think here there are two categories, and like this is not an siRNA question, so neuroscience it spans across targets and sites of action within the brain and targets and sites of action that are outside of the brain. So many of the muscular diseases are outside of the brain. We are seeing several examples, including some from us in the neuromuscular space that are systemically delivered, either by IV or by sub-Q. We've seen FABs, we've seen IgGs and fatty acids. Um, those are all delivered standard dosing routes, IV and sub-Q. There are also examples, including from us, of in-brain targets. So we have a drug in the clinic now that is targeting tau, and that's an IT delivery.
Mhm.
So I think it is just like with any class of drug, if you're crossing the blood-brain barrier, you are going to have to get tricky.
Great. So I think we've reached the top of the hour. If we didn't get to your questions, we'll forward those to Jeff for potential follow-up in the future. I'd like to thank our speaker, Jeff Kearns, for joining us and giving us an excellent presentation today. Please check the ROSA website for upcoming webinars. They are usually once a month. And thank you for joining us, and we'll see you next time. Bye-bye.
Thank you, everyone.